This is the video podcast for Chapter 21 on ecohydraulics, and this is the last chapter of the video podcast for this course. And for this video podcast, I really want to switch gears and do something that's a little bit different and a little bit fun compared to what we've already been looking at. For the vast majority of this course, we've really been focused on the water balance, and that really is the quintessential aspect of hydrology as a science. In many ways, it's really just a starting point, though, for hydrology. There's a lot more that we can do than just, track a water balance. And I've tried to use this course to show how water can interact with different biological factors, primarily focusing on vegetation, though, as a good starting point. However, we have done things in the course like look at the natural flow regime or look at processes in wetlands and floodplains, like systems where the landscape itself is much more dynamic compared to just forests on a hillside. Well, what I want to do here is move beyond vegetation, although a lot of this could be applied to vegetation too, and look specifically at hydraulics and the relationship between physical factors and biological factors in the aquatic environment. So remember that hydrology means taking rainfall and turning it to runoff. Essentially, it's the water balance, right? Where is water stored and where does it flow? And usually, at most scales, that's going to mean taking rain and turning it into runoff. In contrast to that, hydraulics is the study of the motion of water. So we're talking about depths, velocity, shear stress, and then anything we can derive from that. And this concept of ecohydraulics is about looking at the role of flow and these quantitative metrics of flow and how they interact with or are used by different organisms. Well, when we think about the aquatic environment, of course, there's lakes, there's wetlands, there's rivers, there's the coastal zone, estuaries. There's a whole bunch of different ones. What I'm going to use in this case is the topic of rivers, because within the terrestrial environment, rivers are certainly very important to humans, perhaps, in my view, I think more so than others, because first of all, the vast majority of human populations are dependent on rivers for water supply and for proximity of water for part of the way of life that they have. I've showed some of the population demography in the first urban lecture. Far more people live near coasts than far inland, but even still, they tend to be located at places where rivers meet the sea, so there is that riverine component. When you look at the full range of rivers, though, from mountains to foothills, piedmonts, rangelands, and then into lowland environments or deserts, there's just such a tremendous diversity for what rivers can look like. Compared to the HGM classification of wetlands, attempts to classify rivers require an even broader perspective in many regards. It's very, very challenging. So when you ask the question of, how are we going to assess rivers, whether that's from a scientific perspective or a management perspective, it's really challenging to come up with one approach that's going to address all of these different settings. So let's begin by taking an ecological perspective. We can consider the concept of ecological functions. So functions are the ways in which organisms interact with each other and the environment around them. And we can do this from either an organism-centric perspective or from an environment-centric perspective. From a traditional ecological approach, that would be more organism-centric, where we would say, okay, what is the status of an individual? You could look at health metrics for individual organisms and then see how well they're doing. You could then move to the population level, and we could look at population level metrics such as abundance of individuals in the population. You could look at reproduction processes for the population. You could look at genetic and phenotypic diversity within the species itself, usually at the population level, because a population might be in a geographically limited area, and so therefore, what is the diversity that you see of that population within that area and then between that area and other regions where the same species is present. Then you can move up to the community level and look at how different organisms are interacting with each other, whether it's different kinds of aquatic organisms or aquatic organisms interacting with riparian organisms or birds or whatever it is. We consider that set of interactions as part of the food web. We can look at the structure of the community. We can look at the different levels of organisms in the community. And we could ask specific questions related to human activities or dynamic changes, such as the issue of native versus non-native species, both through natural migrations as well as human-induced ones. From an environment-centric perspective, we don't worry as much about the details of what's going on to those organisms as we worry about focusing on creating the physical milieu within which those organisms are going to live. And so we could begin with something like the natural flow regime and characterize the aspects of that regime. And I encourage you can go back and look to see what the 5 components of the flow regime were. But besides just the flow regime, there are other natural regimes part of the physical system. Temperature is something that you looked at with the wetlands homework, so that would be one example. Sediment is independently supplied to rivers through processes such as landslides, gullying, sheet wash erosion, rills, channel erosion, bank erosion. So all those sources of sediment then are moving through the environment in collaboration with water. And so just as there would be a natural flow regime, you could imagine that there would also be a natural sediment regime. We just don't have as much data about sediment because we don't have a national infrastructure for monitoring it the way we do for flow. And that could be done, if it can be done for sediment, of course, it could be done with any constituent. It could be the carbon cycle, the nitrogen cycle, phosphorus cycle, sulfur cycle. It could be related to chemical transformations and reactions. So there's all those kinds of regimes. In addition to that, we can think of processes such as disturbances, whether they're human-induced disturbance regimes or natural periodic events that take place. And of course, the natural flow regime could be part of that, but we could also have a natural disturbance regime if we're talking about things like hurricanes and forest fires and gap creations in forests and whatever. The landform itself is dynamic. So, when we moved into wetlands and floodplains, we really had to spend a lot of time on the hydrology talking about that dynamism of the landscape. There are migration pathways that are required for the populations of different species, but the pathways themselves are really the physical conditions, what we might, they're a type of habitat, spatially connected habitats, and so what are the nature of those pathways? And then finally, there's a variety of spatial scales of features that exist in rivers, which could get all the way down to saying, an assemblage of boulders or wood structures that might be present. Okay, so we have a structure that could be present for populations and communities in terms of the abundance and distribution of those organisms. And we have different functions that could be taking place, like competition, predation, birth, death, migration, and so forth. And so what we need to do when we think about eco-hydraulic analysis is we're going to want to evaluate these structures, functions, and interactions at the different spatial scale, well, not spatial, but ecological scales of individual population and community. And then we're also going to want to characterize the abiotic, biotic linkages relevant to each scale. Of course, if you think about the totality of a setting that you might be looking at and the community that's there, it could be quite a daunting task to go after the whole thing comprehensively. There are times when that's warranted, but there are other times when it may be enough to identify what we could call an ecological indicator. This might be a subset of functions or aspects of the structure that can diagnose the overall status of the system without necessarily having to look at the thing in its entirety in detail. For example, just as an individual human being, if I want to know about myself, I can go get a blood test done, and there's a suite of metrics that can be done, and for more money, more things can be done. But you can get a pretty good sense from a blood test about a lot of aspects of your individual health. Or you could just measure your weight, and a lot of things correlate to your weight or BMI. So those are diagnostic indicators, but of course maybe something in your big toe has a problem that won't be ascertained that way. So an ecological indicator really can be any kind of metric that you think is telling you something that's correlated or connected, or it's an indicator of something else. And hopefully the more other things that it's connected to, then the more useful it is and the more economical it is to really target that indicator. Another thing, just in terms of the basic jargon of what we're talking about here, is the idea of physical habitat. Physical habitat is a little bit different from just habitat, in that sometimes we think of habitat more in the sense of like a niche. So it's the attributes of something, but not necessarily its physical, or not necessarily its geographical coordinates. Okay, and so with physical habitat, well, it's that same idea. It's that we want to know what are the physical attributes that are what are necessary, without necessarily saying, it's here or there, or what is the exact geographic location. And we tend to look at these metrics at two different spatial scales. One is the point scale. So if I look at this, say like a 1 meter by 1 meter square in the ground, that's what we would call microhabitat. Whereas if I'm looking at something that's about half a channel width to 10 times channel width, and there's some flexibility there. The slide here says 0.1 to 10, but basically bigger than a square meter, but only up to about 10 times the if you look at the length of a river, we can measure that in multiples of channel width. And so 10 times when the river is 10 times as long as it is wide, like that small section of river would be like the biggest size that we would call something like a mesohabitat. So mesohabitat is more like a patch. Like on the landscape, we've talked about patches where an individual wetland might be a patch, or maybe a clear-cut section up in the mountains might be a little patch. And in the same way, this is in a river context, a patch would be reference to the scale of the channel width. So if a stream is only 1 meter wide, then that patch might be half a meter to 10 meters long. On the other hand, if it's 100 meters wide, then it could be up to 1 kilometer long. And then the habitat metrics that we would be measuring physically tend to be things like depth, velocity, substrate, and temperature. There's also this idea of cover. So cover are physical attributes that can provide shade, or hiding, or what we could call refugia, where organisms are able to get out of view of predators or competitors. Okay, so here's just a photo of a river that I took, and you can see, if you really just sit here and ponder it for a minute, there's a lot of different kinds of places that an organism can go. Some organisms might want to be out here in the very fast water, enjoying what we might call a feeding station, where as stuff is flowing down the river, then they're going to be getting that food right off the bat. They might like to get in down under the fast water. Maybe they can hide from others by being in the fast flow, but right on the bed under it. Of course, you have this sort of swampy area off on the side here, where you have this high productivity taking place. Maybe small organisms can be hiding among that vegetation. For the vegetation itself, there's habitat there, like the water needs to be slow for that to grow. We have wood that's fallen over. You can see a big shady spot there, down wood on the side. You've got a little side channel wrapping around here. There's maybe an island, a lot of shading. There's really a lot of different little spots that are present in here that would represent microhabitat locations that organisms could choose to be. So when you have a river and you want to say something about how well that river is doing, then for the organisms that you're looking at, one of the ways to do that is to specify what we could call physical habitat performance indicators. Performance indicator just means how well are those organisms doing. So I've tried to write these generically, like species X and life stage number whatever it uses most but not all of the suitable microhabitat. Now, why do we have that? That sounds a little weird. Well, of course, if the population is largely absent from the space, um, then we know that it really isn't taking advantage of what's there. Maybe it's new to the area. Maybe it's just being outcompeted or predated on. Those would all be signs that it's not performing well. On the other hand, if the species has completely filled all of the suitable habitat, then probably it's going to start to experience intra-specific competition at a very high level for those resources. And we could also think of that as not ideal. So most of the time we're interested in organisms being abundant, but not so abundant that they're just stepping all over each other and causing a lot of competition problems. Now, of course, that is to some extent a human value. It's a performance indicator relative to biologist expectations. Um, you know, but let's not get into existentialism, I guess. Let's just leave it there. Okay, so another thing we could look at is sufficient quality, number, size, and distribution of habitats, migration corridors between habitats, so that these organisms can do all of the different ecological functions they need to do in each life stage. I think that's pretty self-explanatory. And the last one is a little bit more vague and complex, overarching from a physical perspective, that, when you consider a river setting in which organisms might be, that there's sufficient maintenance of watershed processes and regulatory management practices really throughout the whole watershed to create and maintain suitable physical habitat for all freshwater life stages of relevant species. So this is a little bit of a, all but the kitchen sink sort of thing, where you're saying, the system as itself needs to be physically, chemically doing what it needs to do. And that's not where you can't just look at the population to see that those mechanisms are in place. You really have to get at those physical processes directly. Okay, so moving right along, how do we make an assessment of the status of an individual river? Well, the first thing we have to do is we have to know something about what those organisms want. If we go back to this slide, we have to assess, are they using most but not all of their suitable habitat? And is there sufficient quality, number, size, and distribution of habitats? Well, that presupposes that you know what habitats those organisms want. How do you know that? So this is the idea of habitat suitability. Habitat suitability means what are the areas of the river, what are the types of areas of the river where an organism is going to want to be present. Whether an organism is, mobile, whether, motile, it can get around, go to where it needs to go, or whether it just moves with the flow, whether by choice or by occurrence, it has to get to somewhere that meets its needs. And so that means that as scientists, we need to characterize those needs, and we do that first by doing an assessment of habitat suitability. To do that, the variables, again, are these physical or chemical variables, but here I'm mostly focusing on physical. So we could take it something like depth and velocity. We have to determine what the suitability requirements are. Now this is where things get a little dicey, because there's really two choices you have. Choice one, look at the real world. Go out and make some observations. Presumably, where you find the organisms will be reflective of what they want, right? I mean, after all, if you followed me around to restaurants, you'd figure out which kind of food I like. However, there is one problem with that theory, and that is that no matter what, you have to go somewhere. So if I'm only given a choice of McDonald's, Burger King, Wendy's, and In-N-Out Burger, I will pick one and go eat something. However, if I had a choice of somewhere else, then hey, I would might choose that. So simply looking at where organisms actually go may not be an indicator of what they want, what their real preferences are. It's reflective of the choices they're forced to make in the environment that they're in. If the environment that they're in is pristine and they're genuinely free to choose among all available options to meet their preference, then you're there. Observing what they do is great. On the other hand, if they're in a setting like most settings on Earth, where the environment is degraded, and choice is limited as a result of human impacts, then you have to be careful. And then that's where the determination of habitat suitability might come down to more experimentation, literature review, and expert judgment. Let's assume for a moment that you can go out and directly measure. And so let's say I go out and I'm interested in fish, and we'll just use Chinook salmon as an example because I've done a lot of work on Chinook salmon. So we want to know where Chinook salmon spawn. Well, if I just go out to a river and I measure every location where they spawn, chances are I'm going to get a probability distribution function for where they occur. Just some kind of bell curve or something like that. And here are two from the lower Yuba River in north central California. One is for the velocity, and the other. Forget about the y-axis for the moment, but just imagine that this is a probability distribution, and the area under this curve would sum to 1, or 100% of my observations. So all we do is we just take that distribution, and instead of using the y-axis as the percent occurrence or frequency, we could just change it to be 0 at the bottom and 1 at the top. And then we can have this distribution. Now, there's an entire science to getting habitat suitability curves. There are many, many methods. This is really just to give you the most general idea. A habitat suitability curve is a plot that illustrates the range of preference that an organism exhibits when you go out and collect data for a particular physical or chemical variable. This curve could be based on concept or expert ideas. It doesn't have to be observation-based for the reasons I mentioned earlier. Okay, so once you have those curves, how do you figure out what the physical habitat is in any particular river that you want to look at? Here's the steps. I think you can read through those. I think that will make a lot of sense. So let me just highlight some of the things we need to do. We need to know what the organisms want. We need to then characterize the availability of the physical conditions in the river, and then we have to do a test, like get some observations and see whether our spatial predictions are matched by observational data in our particular river. So I'll walk you through these steps, and things are going to start to get a little bit more complicated now. The first thing, though, it's not particularly complicated, is you have to work with a biologist, you have to be a biologist, you have to know how to observe whatever organism you're interested. In this case, this is a map that shows the lower Yuba River, just one section of it, and every yellow dot here is a location where Chinook salmon adults have created an egg pocket in the riverbed to lay their eggs and have embryos incubate. So this is a case where biologists go down the river on a weekly basis and make these observations. You can see that there are some areas, like in the top center, lots and lots of fish are choosing those areas, and there are other locations just off to the right where there's none. So is the occurrence of this clustering, is this random? Is this behavioral? Or is it a response to physical conditions? Those are pretty much the three main options, and those are what we need to figure out. So the next step is that you have to get for your river the estimation of what the physical variables would be. One way to do that is with computational modeling. And I'm not going to go into this into detail, but this just shows a map of what we call two-dimensional hydrodynamic modeling, which is one of the things that I have expertise in. And so this is just a very simple case. It's actually from a river restoration project. It shows, the top plot shows a map of depth with the whitish gray colors being shallow and the dark blue being deep water, and the scale is given over on the right. The next one, the colors show velocity, with red being low velocity and blue being high velocity. And the arrows here, like these black things, are arrows that show the direction of flow. So you can see as the flow comes up, from the deep section to the shallow, it actually splits and goes around this shallow bar that's in the center of the channel. So if we take this spatial pattern of depth and spatial pattern of velocity, For each cell in this map, we can apply a habitat suitability curve. So we can say, okay, let's say I have a cell that has a velocity of 4, then I'll get a suitability of 0.2. And let's say I have a depth of, I don't know, 2.5, get 0.2. And I can put all that together and create a map on the bottom that combines the depth and velocity attributes to create the hydraulic habitat suitability index. So a habitat suitability curve is what fish want. A habitat suitability index is the specific value that we predict or measure to be present at a specific location in a river. And the bottom plot here provides that map. Because this is a modeled map, it's a prediction, not an observation. And here's another example of a map. This is for spawning rainbow trout up in the mountains in the Sierras at 3 different discharges, 5 CFS, 190, and 3000 CFS. And I like to use this color scheme where blue usually means the highest quality habitat, and then the colors go down to gray being no habitat present. And you can see that at low discharge, there's lots of places here for those fish to hang out. But by the time you get up to 3000 CFS, the river's just roaring, and there's not a lot of places for these fish to survive. So this is how we make a prediction, a spatial map, for example, of where we think the fish would like to be. The next thing we'd like to do is determine if that prediction is accurate. And that's a process that we call bioverification. So bioverification is a test of the combined predictions, combined meaning hydraulic prediction and habitat suitability prediction, all together to give us that hydraulic habitat suitability index. So a bioverified model is one that is fairly risky. Like, we have to take chances, make predictions that are as specific as possible. How do we do that? We're going to use a metric that I'm going to call the electivity index. So what does this mean? Okay. Well, first, just look at this little map on the right. Imagine we're up into these different patches. It's pretty much similar to what I've showed here in the bottom, or this one here with these colors. Imagine that I've predicted this spatial pattern of habitat. And then imagine that the stars here indicate the actual occurrences of fish. What makes a good prediction is when the number of stars that are in a patch is disproportionately high compared to the size of the patch. So for example, like, you look at this green patch, there's only 4 stars in it, but there's only 3 green patches and they're all relatively small. So that's a fairly specific prediction. Compare that to the blue. We have a ton of stars in blue, but we also have the most amount of area in blue. You can imagine that if I just took like a bag of stars and I threw all the stars onto that picture, that randomly, the more area of a patch color, the greater the probability that just by random chance alone, stars are going to hit that. So the electivity index is where we take the number of stars and we convert it into percent of total stars, and then we divide that percent of stars by the percent of the available area. So, 72% of the stars are in the blue patches, but only 35% of the area is in that color. So when you divide 72 by 35, you basically get 2. And that means that there's twice as many stars in the blue zone as there is area of blue zone. So that's a pretty strong indication. Like, that means that twice as many are there as you would expect from random chance alone. Okay. Let's see. Okay, so an electivity index greater than 1 indicates that they're preferring to be there more than random chance alone would suggest. Less than 1 indicates that they're there, but it's indistinguishable from random, or in any case they could be there less than random chance would prefer, which means that they're tolerating it, but they may also be avoiding it when those numbers get low enough. If the value is 0, then there's really just no habitat at all. So like in this case, white has no stars, so an EI of 0. That means that they're totally avoiding it, and that's not habitat. So all this functions here. I know math can be scary, but all this is just saying is just take the number of stars, or in this case, salmon reds, divide it by the total number, and that gives you the percent area, like the bed area for unit I divided by the total area, and unit I would be like blue or red or whatever. And then the electivity index is just the percent utilization divided by the percent area. Okay, so let's look at another thing. Let's say that I were to predict that the entire river is in the blue patch type, high quality habitat. Well, if I now take a bag of stars and I throw them out onto that, guess what? Every star falls on the blue. So hey, I'm perfect. I guessed 100% correctly. You see what I'm getting at is that making a prediction, it can be quite easy. For example, if I were to say, I predict that an earthquake is going to happen on planet Earth in the year 2015, chances are my prediction is going to be correct. So why, but what the thing is, no one's going to care about that prediction because it's trivial. If people only care about predictions if you say, I think in 2025, the Washington Redskins will finally win a Super Bowl. That would be a very, very risky prediction to make, I would say. So there's a difference between making a trivial prediction versus making a prediction that people would actually value, that has actual management or scientific implications. Okay, so for example, if blue indicates where the model is telling us there's high quality habitat, and red is indicating where there's bad quality habitat, not only do we have to have a specific prediction, but the specific prediction has to be the right direction. So in the top plot here, if I say that red is terrible, terrible habitat, and all of the organisms go live in the red area, well, guess what? Then I was wrong. So it's not enough to just have a risky prediction. The prediction, actually the content and direction of the prediction has to be right, and that would be what the lower plot with the blue shows. So those are the two main bioverification criteria that I would talk about. One is having a high electivity index, and number two, having the direction of the prediction be consistent with the what you observe. So let me show you a specific example. This is a site that we mapped and modeled back in 2004, and the model then led to this spatial pattern of prediction illustrated by the colors. The black dots are the locations where we actually observe reds in the fall spawning season for Chinook salmon in 2004. And you can see that just visually, a large number of reds occur as patches in areas that are blue. So going back to my first statement a long time ago, well, why are these in patches? Are they in patches by random chance? Are they in patches because just behavior, like, hey, the cool fish went there, so I'm going there too? Or is it a response to the physical environment? What you can see is that these patches do not appear to be random. Just based on a visual assessment of this, not quantitative, but visually, all of the patches are lining up with the highest quality habitat in the blue. Meanwhile, the areas in white that are non-habitat have 0 occurrences, and the areas in red are maybe have 1 or 2, but pretty small. So if I now calculate essentially the electivity index, and this thing, I'm doing an intermediate step. Instead of showing you the electivity index, I'm showing you the percent occurrence and the percent area. So for the other graph, or this thing here, percent UI and percent AI, that's what I'm showing here. So the hatched bars are the percent UI, the black is percent AI, and I is just each one of these habitat quality bins. So if I look at the highest quality habitat, there's almost 60% of the reds occur in an area that has only about 20% of that. So if I divide, like 58 by 20, I get almost 3 for my electivity index. So that's a very, very strong preference. You look at the next step down here in habitat quality, and we do have a preference, but it's only a little bit bigger than 1. Then we look at this, 0.2 to 0.4 bin. We see that we actually have fewer organisms than available space for those organisms, which means that they're slightly avoiding those. And then by the time you get down here to the bottom, I mean, I would really have to say that these are avoided. They can live there, so we've kind of moved towards this term tolerated. Like they will tolerate those areas, but those are not the areas that would be preferred. Their occurrence in these habitat types are dramatically lower than you would expect from random chance alone. For grad students, let me just make one other point, which is hard to understand, but with any given number of points, if I randomly throw out points enough, there is a random chance of getting a distribution that is highly non-random looking. Okay, and so there are tests that we can do to show how much the electivity index has to differ from 1 in order to be highly confident that the distribution is non-random. And that's called a bootstrap test. That's far beyond what I would expect the undergrads to know, but just want to raise that, and I'm happy to discuss that with people later if they want. So what I've shown in summary is that you can, let's just go back here, if you can produce a map of the physical attributes of a river or any aquatic system, then you put that together with a habitat suitability curve, you can get a map of the habitat index, and then we can take the habitat index numbers and bin them to produce these patch objects. Well, the patch objects in this case are based on hydraulics, but they could be based on anything. Just as some other examples, I could just take individual reaches in a GIS and say, okay, well, here's 10 miles. How does this 10 miles compare to the next 10 miles? I could look at scour and deposition. I could say distance from the bank or distance from riparian habitat. I could just look in rectangular boxes down the river. Like, there's any kind of object that you want to make, you could apply the electivity index to, compare the availability of that to the occurrence of organisms in that, and get an assessment. So for example, on the x-axis here, I have different morphological unit types. Morphological units just means like riffle, pool, run, glide, that kind of thing, like the different landforms in a river basically. And these 3 that are shown in yellow have significantly higher electivity indices than the rest, and they're higher than 1, which indicates that they are these actually happen to be preferred with a 95% confidence level to be non-random. And almost 3 times as many choose to be in riffle as would occur by random chance alone. And then these other ones are avoided or tolerated. Okay. I just want to say one last thing here. So no matter what we're doing with the electivity index, like I keep saying, oh, well, this is the preferred thing, preferred. In ecology, there's 2 different concepts we have to be mindful of. One is that the vast majority of organisms of a species in a population are going to make a choice to do something that is the norm. They're gonna follow basically the standard approach for that species, whatever that is. However, there's always a case where there are going to be individuals who will diverge from the norm in their behaviors and choices. I don't know why that is, but that's often the case. Many times first of all, of course, it could just be genetic mutation would be perfectly good reason for that, or phenotypic variation. For whatever reason, that happens, and then those individuals, if they choose something different and they are successful in reproduction, then those choices may carry forward into the next generation if those were genetic variations that would then carry forward. And over time, those could then become important, especially if the fitness criteria for the environment change. So this is getting into some evolutionary ideas. I don't know how many students have studied evolution and other ecological ideas, but the point I'm trying to make is that even though we really want to protect the areas that are heavily used by organisms, we also have to be mindful of the value of diversity in creating a range of choices for organisms to choose. Now, we don't want to take this too far. Sometimes I see ecologists who get very dogmatic on diversity. Diversity can be bad. After all, it's important to have places for homogeneity. We need to, like, for example, we always talk about, oh, reefs are the most diverse places on Earth, and wetlands, oh, we got to save the wetlands. Well, guess what? The number one most endangered environment in the world is probably grasslands. Why? Grasslands are fairly boring and monotonous, right? So we have this real bias, both as scientists and perhaps as environmentalists, to focus on one thing, but there is a place for having sizable patches of homogeneity. So it's important to remember that, but also to balance that. So have places that are diverse and have places that are homogeneous. Okay, I'm pontificating, but I think this is all useful. One of the last ideas I think here is the idea of weighted usable area. So we've gone through this whole process of creating a map like this one, a map that tells us what is the spatial pattern of habitat, and this is all for one flow. Like this is a map for 830 CFS. Well, you can imagine if the flow is less, then the habitat pattern will be different. And so we can make a plot that shows discharge on the x-axis, an area of preferred habitat on the y-axis. If you truly have a census-based map, then this is just area. It's just the area as a function of discharge. Some people call this the flow habitat relation, and it just shows as a function of discharge what's the area of habitat. And typically, of course, if there's no water in the river, there's zero habitat. So that's going to go up. Usually, at some flow there would be a peak, and then at very high flows it might go down. Now, depending on the organism, just like any distribution function, this could be exponential shaped in either direction. It could have any shape. Just depends on the needs of the organism. So it's something that you have to go out and produce. The reason why this is called weighted usable area is because many studies don't actually do a complete census with a 2D model the way I've shown you. They may go out and do a statistical sampling. And so with a statistical sampling, then this is then more of an index where you take the habitat suitability index and you multiply it by the area, and you basically came up with a weighted value here. So this is just like when you calculated the curve number. Your final curve number for each subbasin was a weighted average. So this is like calculating a weighted average for each discharge. Okay, so I want to just end the course with one application. The idea here is that we don't want to just study the environment and do ecohydrology as scientists, but that this really does have implications for how we manage the land and ideally even to rehabilitate the landscape in order to bring back some of the natural functioning that's been lost as a result of human activities. So this is an example of a project that I worked on with a grad student and a couple of undergrads. The picture doesn't actually go with this, but this is a photo of a process of recontouring a river. It happens to be the McCallumney River, whereas on the left are examples of design alternatives for the Tuolumne River below Lewiston Dam. And so we designed 6 different alternatives for the river in this section to be mindful of geomorphic conditions, hydrological processes, and hydraulics, and the relation between flow and habitat. And so this shows Design 1 versus Design 6 for looking at Chinook spawning and Chinook fry. So fry are the smallest life stage of newly born fish. And in this case, I say GHSI because this combines hydraulic, substrate, and hydraulics and substrate, so depth, velocity, and substrate. And this is done at a discharge of 8.5 cubic meters per second, which you can do the conversion if you want to get that in CFS. Flow is from right to left. And you can see that Design 1, which has these bars going across the river, these 3 bars, it has a lot of habitat on those bars for spawning, but not much else. If we look at Design 6, you can see that Design 6 is so I call this Design 1 like a highly accentuated topography, like very accentuated deep pools and shallow riffles. Design 6 is more of a blanket fill. It has gradually varying features in the landscape. And you can see that comparatively, like all of the blue and green indicates that almost the whole river is available as spawning habitat. In contrast to that, when you look at fry habitat, Actually, Design 6 is almost devoid of it. Like, this is just totally useless for rearing, whereas Design 1 does provide some areas for rearing in the sheltered zones behind the bars. So, like, here's this bar going across in the middle, this green and aqua sort of colors back here. That's a place where there's sheltering from the velocities, and so there is some habitat present there. So this just goes to show that when you look at different life stages, the design of a river might actually have very different needs. And so I can look across all 6 designs as well as the pre-project condition and look at the percent of the wetted area in each different habitat quality. Black would be non-habitat, and then blue is the highest quality habitat. So the length of the bar shows the amount of the wetted area that's in that habitat type class, a habitat quality. And you can see that, like there's a lot of Chinook spawning habitat, some steelhead spawning, but not enough. And really look at the difference. Like designs 4, 5, and 6 are very useful for Chinook spawning, and they're pretty good also for steelhead spawning. In contrast to that, like none is particularly great for fry, but 1, 2, and 3 are better. And so what I can do, I could also take, I could look at the area of habitat and divide it by the volume of gravel that I have to add to produce that. So like the topography that is shown here, it's being made, as you can see in the photo, by bringing large amounts of gravel and cobble and filling in the holes in the river to produce this topography. So I could take the surface area of habitat created per volume of gravel added and get a little bit of an efficiency, because gravel would be a surrogate of dollars. The more gravel I have to add, the more fuel I need, and that's an indicator of cost. So you could make an argument that there's an efficiency, and so do we get better efficiency by doing one thing or another? And I'm looking at the gray bars being adding habitat where it didn't exist before, or taking low quality habitat and turning it into high quality habitat. So for Chinook fry, we get the best performance from D1 through 3, the accentuated scenarios, whereas for spawning, we get the best for 4, 5, and 6. And 6 in particular just gives us really by far the best performance for spawning. And same thing for steelhead, it's the best. Steelhead fry is interesting. It actually adds new habitat, but it takes the existing habitat that's there and actually degrades the quality of that habitat. But the fact that we're adding a lot of habitat outweighs the fact that we're degrading the existing habitat. So when you put all those metrics together, you can kind of see, well, okay, what for each of the 4 life stages, what are the benefits in terms of habitat addition efficiency, habitat improvement efficiency, the spatial pattern, and then best overall. And ultimately, because this project is primarily for spawning, then Design 6 was the one that was chosen and was in fact built. And the idea is that there's plenty of fry habitat in other parts of the river downstream. Okay, so this is really just an introduction to these concepts. I wrote a textbook a few years ago called 2D Modeling and Ecohydraulic Analysis that goes into quite a bit of depth of how to do everything that I've described in this video podcast. And periodically I teach a class in ecohydraulics, so if you're a grad student, you can take a look for that. So I appreciate your putting up with a quarter's worth of video podcasts. I hope you found this a useful approach to getting information as opposed to sitting in class and listening to lectures or missing lectures or whatever. So thank you for taking HYD 143.